Good afternoon, everybody. My name is Nicolas Bautista, and I'm currently a post-bachelor student at Charles R. Drew University of Medicine and Science. And today, I'll be presenting the research I have been working on, that is, e-cigarette vaping increases DNA damage and reduces senescence in triple-negative breast cancer. Now, triple-negative breast cancer, or TNBC, tests negative for estrogen receptors, progesterone receptors, and has an excess amount of HER2 protein. The lack of these disruptors does not allow for hormone therapies to be effective, consequently making TNBC the most aggressive form of breast cancer. Also, young African American and Latinx women are more prone to develop TNBC. Currently, ANS, or Electronic Nicotine Delivery System, is very popular and was once considered to be healthier than smoking cigarettes. But our study focuses on the free radicals and harmful chemicals, such as nicotine, that actually increases DNA damage and reduces its cellular senescence response that leads to tumor proliferation. Now, senescence is the process of a cell's deterioration with age, serving as a protection from tumors. So we hypothesized that the nicotine damages this tumor suppressive response and increases the chance for young women to develop TNBC. So now for our experiment, there is a total of 3 different female nude mice. The first group had vape from e-cigarette with nicotine, the second group had vape from e-cigarette without nicotine, and the third group was the control that had no vape exposure. The mice were exposed to vape within the vaping changer, and then, after exposure, the mice were subcutaneously injected with TNBC cell lines MDA MB468 and HCC70 under their mammary pads. After 90 days, the tumors were excised to compare the effects of e-cigarette on tumor growth. Now, we notice on the left-hand side, the cell line MDA MB468, in the top you see that there's just the tumors that did not have nicotine exposure, while the bottom ones were the ones that did have nicotine exposure, and the top towards a control. Overall, you can see that the ones with nicotine exposure are relatively larger than the ones that did not have nicotine exposure. And then we took a further analysis of the tumor volumes, and you can see that the top row right here, the ones that did have nicotine exposure, have a greater volume than the ones that do not. Also, on the HCC70 cell line, again, you see the bottom row with the ones that did have nicotine, the top row without nicotine, and our two control. Instead, you can see that the ones that did have nicotine appear to be larger than the rest. And a further analysis was one where we took the tumor weights of both of these tumors, and we see that the ones without nicotine exposure weigh less than the ones that did have nicotine exposure. Then we did a heat map where we have the 3 groups: our slow-growing tumors, are ones with just e-cigarette vape, and the ones with nicotine exposure. And on the right, you see all these listed genes. The 3 genes that are circled here are CAT5, ASFA1, UBN1, and all these genes have a function of senescence. And so the, the green means that these genes were downregulated, right? Meaning that these genes were not really expressed, while the red means that your genes were upregulated, means that they were highly expressed. So on our in our slow tumor growing groups, we see that they're upregulated, which makes sense, right? Because senescence is happening, so it's protecting the cells from developing tumors. But when you see in the groups that did have nicotine exposure, you see that they're downregulated, meaning that senescence is not occurring in these genes, which actually causes for there to be tumor proliferation. Again, you can see these G genes. In our first group, it's higher gene expression levels than in our last 2 groups for all of them. And there's an overall graph demonstrating again that when there's nicotine involved, these genes are not really highly expressed. So overall, from our data, we want to show that our groups with and without nicotine demonstrate a strong signature of reduced expression of senescence genes. From our data, we hypothesize that e-cigarette increases DNA damage and reduces DNA repair and senescence in triple-negative breast cancer, especially since these vaping products are relatively new. Their health effects are not really studied, and we hope as we continue to do our research that our data and our findings are able to establish more of these effects so we are able to protect more of our young adults. Raw, thank you for coming to my presentation, and I hope the best for all of you. Thank you and take care.